In addition to differences in nutrient inputs, there's also differences in nutrient turnover that are determined by species. Species differences in litter quality magnify site differences in soil fertility. And this is one of the places where I'm going to add on to what the book says and differ a little bit. But it's important to think about some of the differences that occur when we have different species there. And litter quality, as we know, is an important factor for determining decomposition and mineralization rates of nutrients. So when we think about different species, we really want to think about differences in soil fertility and how those two interact to generate differences in litter quality. One of the examples that the book uses in experiments that I've worked on too are comparisons when we have differences in species present on a landscape. And we can test this in common garden studies where we have monocultures of different species. So this is a prairie study. This is from Minnesota, grown on relatively sandy soils. And these right here are monocultures of different species. So they were planted somewhere on the order of 6 years before this photo was taken. And you can see that there's large differences in biomass and structure to these different communities. And all of these have been kept to differences in just single species. So there's one species here, and this is probably a warm season grass, a C4 grass, and this is a nitrogen fixing legume over here. Now, the example that the authors use shows the differences that we get in mineralization rate when we compare monocultures of different species. So these are all different grasses that were planted in monoculture in Minnesota. And here on the x-axis, we have the C to N ratio of the litter. And on the y-axis, we have mineralization rate. So x-axis C to N ratio, low C to N plants, high nitrogen concentrations. And when we look at the mineralization rates in those plots, the mineralization rates are relatively high. Work your way across this gradient of C to N to get into these high C to N plants. And these species that had the highest C to N ratios had the lowest rates of mineralization. And this was used as evidence of some of the feedbacks that occur for having differences in C to N ratio, generating differences in mineralization rates. So we'll often when the authors and other people will talk about effects of species on ecosystem processes, it starts with the assumption that species differ inherently in their C to N ratio, and this causes differences in mineralization rates. And we talked about how having a high C to N ratio is going to increase microbial immobilization, and that's going to decrease net nitrogen mineralization. So in this case, this was a classic example of differences in species traits generating differences in mineralization rates in showing that species can differ in their effects on the ecosystem process. There's a caveat to this approach that I'm going to point out here, and this is part of the reason why I get worried about differentiating species on their effects and their responses to differences in ecosystem processes. So often we'll see as much variation within species as among. And I've worked on those same plots that the generalizations have came from in the previous slide. And we've actually done similar sets of measurements in two different experiments. On the x-axis, I have leaf nitrogen concentration. So we have species that had high nitrogen concentrations and relatively low nitrogen concentrations. And remember, this would have a relatively low C to N ratio. And this would have a relatively high C to N ratio. And on the y-axis, we have leaf longevity. So species that have a low nitrogen concentration tend to have a relatively high leaf longevity. Those species that have a high nitrogen concentration had a really low leaf longevity. They turned over relatively quickly. And this is something that we understood before, and that's nothing new. But when we take a look at this differences among species within and between the two different experiments, here are some of the grasses that were used to generate the previous example. We can one grass, when it's growing in one experiment, can have a relatively high nitrogen concentration and leaves that don't live that long. But in a different experiment, it can have a low nitrogen concentration and have one of the highest leaf longevities. And you can see that there's as much variation within a species as there is among. So this graph, the question that we had on the previous graph, when we looked at C to N ratio, and then we had nitrogen mineralization, and those species that had the highest C to N ratio had the lowest N mineralization. In what case, and what's the causality here? So did the species impact nitrogen mineralization rates that changed the C to N ratio, or did they start out with a different C to N ratio and had that caused differences in mineralization rates? When we think about how plant species traits interact with nutrient supplies, essentially we have a pattern like this. That there are differences in plant nutrient concentrations, and think of this as something like %N or C to N ratio, and soil N availability. And determination of some plant traits requires understanding the growth and the feedbacks to nutrient cycling. So for all intents and purposes, when you start up a monoculture and plants are growing, that they all have relatively high nitrogen concentrations. There's a lot of mineralization that occurs. It's a disturbed system, for example. And some species are going to grow faster than others and have different effects on soil nitrogen concentrations. And that's going to feed back to change in plant C to N ratios or N concentrations. So what we do is we're going to start, if this is going to be, let's make this %N, all the species might start out here, and then some of them shift over time, something like that, and others shift over time to having even lower nitrogen concentrations and differences in leaf longevity. So in this case, we can talk about species differ in their nitrogen concentrations and having an impact on soil nitrogen availability, but really it's a very interactive process and it develops over time. And we can't separate out whether species actually differ in their nitrogen concentrations or that the way that they impact nitrogen availability. We can't separate out effect from response that easily. So in summary, that effect and response transition or difference between species is actually hard to quantify for some of these different factors. But we do know that we can develop differences in nitrogen concentrations, and there'd be differences in the impact on nitrogen availability. But what actually causes that is a different question.